Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
I think I'm ready to 30, right? Maybe we can start. Participants only 7. Yeah, we have already. Hopefully, SOD can remind the staff who want to join. Yeah, so I already share the link through the WhatsApp group. So I would like will be very appreciated if you can share to your staff. Okay, anyway, I think it's already 2:30, so we can start ya. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Let's begin our talk today. Season of Quran Al-Fatihah. Welcome to the KOP Tarik. This is the series 3 already. Yeah, so it is a platform for us to communicate and share the research experience among KOP academic staff. So, and also thank you to all of you having some time to attend this talk. Hopefully it can benefit all of us. Okay, so for today we have two speakers. One is Dr. Awis, yeah, and the speaker number 2 is Dr. Muhammad Taufik, yeah. For Dr. Awis, he has some time already in study. This is the your study during PhD, right, Dr. Awis? So this is a very good sharing today about what he performed last during his PhD study. Maybe also some data also during his time as lecturer in IUM. Okay, without further ado, I would like to welcome Dr. Awis to present.
Thank you, Dr. Alfi.
Okay. Let me share the screen. I already share to you. You can share.
Alamak, salah buka lah. Okay, bismillahirrahmanirrahim. Okay, assalamualaikum and good afternoon, everyone. So thank you for the SOR office for inviting me to share a bit of my knowledge, which is not that much compared to all of you, I think. Okay, the topic that I choose for this afternoon is pharmaceutical material science, the road less traveled. So the reason why I choose this topic is because I think most of people not really familiar with the term of the pharmaceutical material science. So, okay. So what is the pharmaceutical material science? So it's the application of the fundamental concept in the physical sciences to the challenges of understanding the behavior of soft, mostly organic, crystalline, and amorphous material of relevant to the pharmaceutical industry. So it's concerned with the connecting the phenomena occurring on the molecular scale, such as the crystallization and polymerization to metric of macroscopic performance, such as hydration rate, mechanical strength, and their consequences for industrial processes such as powder flow and the compaction. So how did I end up with the this topic? So actually during my research in when I did my PhD in UCL UK, our research group, the focus of our research group is basically on the thermal analysis. So basically what we did is most of the study is involving the thermal analysis. It means to improve the solubility of the sample, or maybe you can going to combine or the sample or encapsulate the sample into like a microfiber, that kind of things, and then we end up testing using the DSC, AFM, TTM, that kind of things. So during that time, one of my friend, he did a PhD as well, but in different university in London. He came to my lab, and then after, when he came there, so I just show him around. So I showed him, okay, this is the instrument that we use, and then this is what I'm doing currently, what I'm doing for my PhD, etc., etc. And then he noticed that they said that my research is basically almost similar with him, and his research is actually in material science, because he's an expert in material science. And then he said that all the instrument that I use is basically been used in the material science. So that's how I know that, okay, my research is actually more like a sort of like a sub category of the drug delivery system. So it's more focused on the material properties of the material the properties of the material, rather than like the normal pharmaceutical formulation. So in this talk, I'm going to share you two of my studies. So the first one is the one that I did during my PhD, and the second one is been done by my RP student actually. When I already here. So this one is the one that I did during my PhD. So the title of the study is the investigation of the nanoscale surface analysis on different sample surface smoothness using the transition temperature microscopy, or the TTM. So as you can see here, this machine is what we call a TTM. It's just a small machine, and the thing is, not many people, even in the world, not many people have this machine. So and then it's quite expensive as well. In this study, basically, I'm using the TTM to measure the transition temperature of 3 different polymer. This is HPMC, PVP, and PVP90F, and the PEC 6000 with the different surface smoothness. So in this case, I choose a mini tablet and the firm surface. The mini tablet, because it's basically direct compression, so the surface can be rough compared to the firm, which is have a very smooth surface. And so before I go deeper into explaining about the study, so I need to explain how this machine works. So here you can see there is the tip that attached to the TTM. So when you run the sample, when you run the machine, so the tip or the probe will lower down, and then so it's going to touch the surface of the sample. And at this point, it's going to heat up the sample. And then once they have the sample, they will measure what is the transition temperature of this point here. And then as you can see here, at this point, once they heat up, and then when the surface is softened, it's going to drop. And then they're going to measure the temperature. In this case, the temperature is around 207.9 degrees Celsius. So you're going to repeat the same process at a different point here. Like this one, for example, this one is a 100 micrometer times 100 micrometer. It's a 100 micrometer square of a area of the sample. So here, I think roughly they have like roughly 121 points. So after we're done at this point, and then they're going to lift up, and then it's going to lowering down the probe, and then going to test this transition temperature of this area. So they're going to repeat all these points, and then after that, it's going to make it into a map of the temperature of each of the surface area. So at this point, you can get the idea what is the material that you have on the surface of the sample that you have. Because normally on the sample of you have like normally you have an API, you have the excipient, for example, so they can map where is this excipient and where is this API on the surface of your sample. So in my study, basically, I prepared 6 different mini tablets actually, based on two different particle size, which I categorize as a smaller sieve fraction and the larger sieve fraction. So I make it into a mini tablet, and then I test it on the surface of the cap, or on the top of the tablet, and then the other one is on the on the side of the tablet or on the band of the tablet. So before I start the study, I run the sample using the MTDAC, which is the modulated temperature differential scanning calorimetry, just to test if what is the actual reading of the sample that I use. So this one is the value that I got from the reported value from this article, and then this one is the one that I got from the MTDAC analysis. So as you can see here, all the reading is actually within is agreed with the one that been reported by other researcher here. Okay, so this is the TTM map and histogram that I got from the smaller particle size of the HPMC. As you can see here, based on this one, you can see there are a few different there are a variable of transition temperature on this surface area. Bear in mind, this one only consists of HPMC. So by right, they only have one transition temperature. But here you can see there is a scattered here. It sounds like there are a lot of different material here. And then for the HPMC, like as it's mentioned here, the actual reading should be around 190 degree. But here you can see you can find the result. There are 6 points showed a 0 degree result, and then there are several here and here, so it's scattered here. And then so based on the MTDAC data, we expect the color of the grid is going to be green or yellow, because green and yellow should be around this region, so around 190 degrees. So it should be green and yellow only. But from the map, you can see there are a lot of different colors. This means it shows different temperature of that range from 25 to 250 degrees. And for the smaller particle size of the tablet, it shows temperature between 150 to 250, whereas the maximum occurrence is between around 200 degrees Celsius. For the bigger particle size, you can see, actually I didn't put the diagram here, but it's the range of the temperature is between 260 and 220 and 260 degree, with no value near to the 200 degree. So the result shows that the smaller particle size is a better agreement to the expected value, but still we can see from the histogram and the color image that there are 0 degree temperature showed in the sample, which is they shouldn't, because the temperature that we run is between 25 to 250 degree. So overall, approximately half of the individual reading were incorrect based on the MTDAC data that we discussed earlier. And so this one is the result from the mini tablet of the paravinyl pyrrolidone 90F, PVP 90F. The same case with the this one it showed the same pattern with the HPMC just now. You can see here there are a lot of different transition temperature here, even though it's just only one compound or one polymer. And then if you look at the histogram here, there are a few points here showed like actually there is a between 225 and 250. Basically, they detect a lot of different what call it transition temperature between this range. So again, it should we expect it should show a green color, because based on the MTDAC, the result show that the transition temperature is around 180 degree. And then again, similar with the HPMC, we notice there are 0 degree reading as well. And then there is no obvious peak on the histogram. You can see here it's quite scattered compared to the one that here. You can see that it is sort of like a focusing on this area, but for the PVP is quite scattered. And then approximately half of the reading were incorrect based on the MTDAC data. It means it's not near the 180 degree. And the TTM map of the mini tablet on the band or on the side of the tablet is significantly poorer compared to the cap of the tablet. And then we move to the polyethylene glycol 6000. So this one, as you can see here, it show like it's quite a uniform color. It means the show and then if you look at the histogram here, the melting point or the the transition temperature here is focusing on around this region. It means it's around 33 degrees Celsius here. So it means that it should a consistent value throughout the surface of the sample that we analyze. And then so the value of the melting point that we get is around 31 to 34 degree. Even though it's like it is lower compared to the one that the actual value, which is for the PEG, it should be around 60 degree. But there is a explanation about the one, which I'm going to tell you later on. And you can see there is no different in the TTM result compared to the rest of the sample. It means when we test it on the cap, on the side of the tablet, and different particle size, all of them showed a different a similar result. So after we got this result, so we try to figure out what is actually the problem, what is the possible problem that caused this thing to happen. So we hypothesize is because of the surface of the sample. As you can see here, this one is the SEM image of the HPMC, PVP, and the PEC. For the PVP especially, you can see there is a lot of gap here. You can see there's a particle that been meshed together, been compressed together. And it doesn't really gel well compared to the PEC 6000 here. You can see it's quite smooth. It's less crack, and even the crack is quite small. And then this one is quite smooth for the HPMC. But still, you can see there is a you can see kind of obvious particle here and here. So based on this picture, we hypothesize that there are three possible ways of how the probe contacting the surface of the sample. The first one, of course, is going to contact directly on the surface of the sample. So when this thing happens, it will show you a proper reading. It means the actual reading, the one that the accurate one. And then the second option, the second possible way is basically they're going to land at the edge of the sample. For example, like this one, at the edge of this area, for example. So when this thing happens, it might show you like an inaccurate reading. And then the third one is, of course, it's going to land at the between the gap of the sample, for example, like here. When this thing happen, the probe is going to lowering down at the until a certain limit, and after that it's going to hit up this area. So once they didn't notice anything, so it's going to hit up from 25 to 250 degree. So if they didn't notice anything, they're just going to record it as at the 250 degree or 0 degree. So because of that, we start to prepare a film, because the film have a smooth surface. So in theory, if you prepare a film, then you should get a uniform result based on the TTM. So when we run the TTM on the HPMC film, we noticed that we still found that there are a few different temperature here. This one, there's a purple, and then the rest is a red, and then there is an outlier here, blue color. And then when we look at the histogram here, you can see 90 point is basically at a 0 degree, and then the rest is at the 250 degree. So based on the TTM map and the histogram of the HPMC film, it showed, like I said just now, it showed that 90 out of 121 points were recorded as a 0 degree, while the rest showed the maximum temperature transition, which is 250 degree. And it shows a similar pattern compared to the tablets, but this one is more extreme, because the previous one, there's still a few point in between 0 and 250 degree, but here you can see it's more extreme. There's only it's at the 0 or 250 degree. And so since the surface of this film is basically smooth, so there is no crack, so it is impossible for us to say that the reading obtained from this TTM is because of the improper contact between the probe and the sample. So what they did is, when they couldn't determine what is the actual transition temperature of that area, they're going to record it either 250 degree or 0 degree. So the probable explanation behind this behavior is because HPMC have a very indistinct, a very small glass transition. Even when you run it using the DSC, sometimes it's quite hard for you to find the transition temperature of the HPMC. This one is been reported by the McPhilip. They found when they run the HPMC using the MTDSC, and then so he concluded that we need to use a large sample size in order for us to measure this transition temperature of the HPMC. So in case of the TTM, because we are looking at very small amount of the HPMC because it's just at the tip of the probe, which is the size of the tip is less than 30 nanometer. So it make it almost impossible for the TTM to measure the glass transition temperature of the HPMC. And then we move to the PVP90F film. As you can see here, it shows like a very nice green color here. And then when we look at the histogram here, most of the temperature is show like a transition temperature at around 186 degrees Celsius here. So this one is confirmed the theory that we have is made before this, that they cannot show the actual reading because of there is an improper contact between the probe and the surface of the sample. And then based on the MTDSC result, the TG of the PVP is actually around 178 degree, but for the TTM it shows is quite high, 186 degree. But this one is quite common because based on the article by Royal et al. In 2001, he found that the TG value observed with the thermal probe movement measurements such as TTM and other thermal measurements such as the MTDSC can differ up to 20 degree. So this one is because of the probe measuring the softening process of the polymer rather than its actual TG point. So that's why there is a difference in temperature compared to TTM and the DSC. However, the result showed it showed a consistent value of around 186 degree. So hence with the TTM again, like I said, because the TTM result on the PVP film, it shows a very consistent, which is because of the surface of the film is very smooth. So it's basically it's justify our hypothesis before. And when we move to the PAC 6000 film, and you can see here there is a similar profile showed compared to the one that we have in the tablet. So basically all of them is roughly around the purple and bluish color. And then if you look here, it sounds like it's scattered, but this one is because the scale is quite small. This one, the scale is just between 0 to 40 degree. So that's why you look at it's a bit scattered. But actually, if you stretch the scale here to 250, it's going to show you a very nice peak. And then so this one, the value is around 35 degree. And as I said, because based on the PAC 6000 tablets, it's already showed a smooth surface. So that's why the result that we got from the PAC 6000 tablet and the film is similar. So that is the study that one of the study that I done during my PhD. As I said before, during at the beginning of the study, the importance of the study is actually because let's say we prepare the film, and then so we want to see how well our API or the active ingredient distributed across the surface of the film. So that's why we're going to we can use the TTM, because if you use the DSC, we can only get the transition temperature of the bulk of the sample. It means we cannot really say how well the API or the active ingredient that we put in the film scattered throughout the surface of the film. So moving on, so this one is the study done by my RP student. So this one is basically we want to improve the solubility of the ibuprofen, because ibuprofen is fall under class 2, which is mean it have poor solubility but good permeability. So that's why we use this one. And then so what we did is we're going to encapsulate the ibuprofen in the microfiber. So how we produce it is basically we use the candy floss machine, and I still have it in my lab, this machine. So we during this study, what we did is we play around with the ratio of the ibuprofen and the sugar. Is between 5% of the ibuprofen to 20% of the ibuprofen, just to see how much that the maximum amount that we can encapsulate using this technique. And then for the sugar, we use 3 different type of sugar. There is a glucose, fructose, and the sucrose. So from the result, the first one we try to coat these 3. We try to increase the ratio of the drug to the sugar ratio. But the only ratio that we managed to spun is just 5% of the ibuprofen. So we proceed with the 5% of the ibuprofen, and then we did the solubility test on the pure ibuprofen, and then we got the solubility of the pure ibuprofen is 0.0233 milligram per mil. So it's similar to the one that we find in the based on the other study, which is 0.021 milligram per mil. So after we spun we encapsulate the tablet, or the drug in the sugar, and then so we found that there is the improvement is basically 100%, is double of the solubility compared to the solubility of the ibuprofen as a pure compound. As you can see here, for the sucrose, the solubility improved from 0.023 to 0.042, and for the fructose, it shows around the same, 0.044, and for the glucose is around 0.042. It's quite close to each other, and then when you compare it to this one, it shows like a very significant improvement in terms of the solubility of the sample. And then after that, we run using the JSC result to see if there is any changes in terms of the melting or the TG of the sample. Because when you mix the drug with the material, there is tendency for, especially for the sugar, there is tendency of the transition temperature to be shifted compared to the actual or the original melting point. So if you can see here, for the ibuprofen, the melting point is around 74 degrees Celsius. For the glucose is 155, fructose 122 degree, and sucrose 197 degree. When we combine the ibuprofen and the glucose, you can see the ibuprofen melting point shifted up a bit to 80 degree, and for the glucose is shifting down a bit to 149. And for the fructose, the melting point is basically similar to the one that with the ibuprofen, but we cannot see the melting point of the fructose. At this point, we speculate is because at this point, when mixed with the ibuprofen, the melting point is become too small for it to be detected. And then for the ibuprofen and sucrose, the melting point of the ibuprofen shifted down to 69 degree, and for the sucrose is shifted up to 157 degree. And so we run it using the XCR to see the stability of the sample. Actually, it's quite hard for you to see here, but for the ibuprofen, they have like a very what we call is a significant peak around 1,710 here, compared to the rest of the sugar, because the rest of the sugar they don't have that peak around that region. And as you can see here, you can see there is a peak here for each of for every of the every of the mixture between the ibuprofen and the sugar. And during this one, like I said before, the functional group of the ibuprofen is the carbonic group that is very strong to compare with the other band in the ibuprofen spectrum. And then the wavelength of the carboxyl group can be seen around 1,710 on the ibuprofen spectrum. And this peak is can be clearly seen in the each of the mixture of the sugar between the sugar and the ibuprofen, which is indicate that the functional group of the ibuprofen exists in the fiber, thus suggesting that there is no interaction between the drug and the sugar. So that is the two of the study that that I've done myself, and the one that been done by my student under my supervision. And so, what is the latest update based on similar study that I'm currently doing now, which is in the pharmaceutical material science. So currently I'm co-supervised two master students with Dr. Solah on the 3D printed tablets. So this one it can be categorized under the pharmaceutical material science as well, because basically we are combining the drug and the excipient, and where we're going to look at the if there is any changes in terms of the melting point, etc., etc. And then I'm also collaborating with Dr. Muzammil from the IUITM, Dr. Juzaili from USM, and Dr. Ashura from the Institute of Medical Research on the conductive polymer. For this one is the current collaboration is more on the material science actually, because we already published several articles on the usage of the incorporating the conductive polymer on the what do you call it, on the clots. So because of the conductive polymer, they basically is a polymer that have a current, a small current, and then so we are planning to incorporate this one in for the pharmaceutical formulation as well, where we're going to use this conducting polymer for the wound healing treatment, because of the small current that they produce with the polymer, that it can help with the wound healing generating to improve the wound of the patient. Okay, I think that's all. This one is my collaborator. Thank you.
Thank you, Dr. Awis. It's very interesting finding. And yes, of course, is many people would like to apply your knowledge for the research in our faculty. Okay, any questions from the floor? Any questions?
Assalamualaikum, Dr. Awis.
Assalamualaikum.
Solah here. I joined a bit late, sorry. So the TTM map, that one we want to know the distribution of the drugs inside the
the concept of the TTM is basically just like a is a minuscule of the DAC. It means the DAC in a small is on the small scale. So because for the DAC, basically we're going to measure the transition temperature of the sample as a bulk. But for this one, let's say this one is a firm, we're going to detect at this one point here only. So let's say on this firm, we have like an API, you have the polymer, so they can map what is the material that you have across the surface of the sample that you have here.
Is it like Raman? What is it?
It's sort of like Raman. Yeah, the theory is almost like a Raman.
I see. Okay. Do we have here in Malaysia?
I don't think we have it because the probe is quite expensive as well. The probe of one box of the probe containing 5 probes, and the cost is £1,200, and it's easily broken because it's very fragile. So and
Okay, thank you.
so it's very expensive. I'm not sure. I don't think that we have it here in Malaysia.
Okay.
Any other questions? Any other questions from Tech?
Assalamualaikum, Travis.
Salam, Prof. Tahir.
Very interesting. Still, I'm interested with your title. I think your first slide. That one, there wrote less travel. Maksudnya, this one is regard to the pre-formulation, right?
Because
Because we know that pre-formulation is very important in pharmaceutical development. Because there are some to analyze and need to measure before active drug to be formulated. So I think with the method you have been studying, is it much faster or?
putus-putus. Can you repeat the question again?
Boleh dengar? I think line is not.
Putus-putus.
Level 5 macam tu kot.
Line terkena COVID.
Very good in my office.
Putus-putus. Pak Tahir sebut putus-putus pun putus juga.
Putus-putus ya, terdengar. Terputus, saya dengar. Atau Prof Tahir boleh type je. Tak apa, ini je, apa namanya? Type je, Prof Tahir. Boleh ini, boleh type dia punya questions.
Tak perlu Starbucks kot, touch cukup lah.
Oh, Starbucks je. Okay lah. Okay, any other questions? We still have time. Okay, Dr. Awis, I have one question regarding the TTM result. One slide that is because in your result you show no the curve.
The curve.
I think after TTM result you have the curve, right? So this one. So you when you have this curve, so you only check the peak, right?
Yes.
Not the slope.
Yes, the peak.
Oh, only the peak.
This one is actually when the probe touching the surface of the sample. So the probe they start going to heat up the sample and until the sample is softened or melt. So one is melted. So because this one is based on the force of the on the force of the tip as well. So when you heat up the sample here, and then when they soften, it start to drop. So when they start to drop, and so the probe will stop. The machine will stop hitting the probe. So and then they're going to register the softened part as the melting or the TG of the sample.
So meaning that they will record the peak regardless the time required.
Yes. Yes.
Okay, I understand. Okay, any more questions regarding this? Otherwise, we can go to next speaker, Dr. Muhammad Taufik. So thank you, Dr. Awis,
Okay, thank you everyone.
very much. So we will have the second speaker, Dr. Muhammad Taufik. The title is also regarding the nanoparticles. So the microfluidics fabrications, nanoparticles for the mass. Okay, you are welcome, Dr. Taufik.
Thank you, Dr. Alfie.
Yeah.
Share my screen first.
Okay, you can share. I have already shared to everyone.
This one. Okay, is it
Yes.
visible now?
Yeah, I can see.
Okay. Right. So thank you very much to Dr. Alfie, and thank you also to all the attendants for all people who attend today. Okay, so it's been a while since I talked about my PhD project. So basically, this is what I did during my PhD. Then in the subsequent years, I haven't had a chance to further develop the project, basically, right? But then again, we'll talk about that. Anyway, what I want to talk about is about nanoparticles, so liposomes, everything. But this is something that I would say it's very common. People know about this. There are commercial products that have been in market, approved to be used, and everything. And even our Pfizer vaccine is also liposomes, so liposomal mRNA. But the issue with liposomes is always about price. When I went into this research project, my intended outcome is to find an old drugs, so old out of patent drugs, and repackage them so that we can make it cheaper and more efficacious to be used to the patient. So one of the methods to do this is by encapsulating them and give them a proper targeting system and things like that, so that we can send these drugs, this old and it's much more dangerous and have higher side effect, to where it's needed. So you can see that in encapsulating all of this, it can protect the drug itself from the harsh environment outside, and it can send it to where it's needed, and also protect those that have no business in interacting with the drug. Right. So I chose liposome because it's quite ubiquitous. Everywhere we can see and use liposomes. They are the constituent lipids are easy to find, and if we go for newer types of nanoparticles like niosome, for example, they are more, you can say at this stage, experimental and not very widely used. Okay, but they have promise. So today the talk will be about these types of nanoparticles. Doesn't matter whether it's liposome, polymer, or niosome. Right? So why we are using microfluidics? Now, the interesting thing about microfluidics is that you can look at all other methods of producing nanoparticles. The easiest way, the tested and true method is the thin film hydration, of which we dilute the lipid inside a round bottom flask, and then we dry it up. We dry all the ethanol or whatever lipid diluent that we use, and then we put in the aqueous part of it. The aqueous part usually contains the drug in this case. So basically, it will rehydrate the film of lipid, and then it will encapsulate the aqueous drug within that, within those layers. So basically, it becomes liposome in that sense. But it will form liposomes of various sizes, so we still have to do some post-processing. We can do extrusion, for example. So to make it, or sonication, we can make it smaller in size. So these things, while it's good in terms of lab scale and everything, but if we think about larger scale, it's not really feasible. So my focus during my study is that I want to find something that we can immediately put out and commercialize it, or something that we can develop and then we can immediately scale it up without compromising in terms of the characteristic of the liposome itself. Now, the microfluidics method is basically a mixing method, but it's done in a very small channel. Okay. And then it will form a laminar flow, which I will show in the next slide. In this case, it will mitigate the limitation of the conventional method. Basically, we can make it continuous stream of produced liposomes as long as we feed in the aqueous and the lipid phase, which is in this one. So this one is usually something that we can see like a conventional design of microfluidics. So we have in one channel the lipids dissolved in alcohol, for example, or any other non-aqueous diluent, okay? And then we have either tangentially or from different angle, 30-degree angle or something, an intersecting stream of aqueous phase. So this aqueous phase usually contains the drug itself. Now, at the interface layer between these two streams, you can see at the topmost part of it, at a critical concentration of these two, the liposome will spontaneously self-assemble and then will entrap the aqueous phase in it as well. So they will entrap the drug in it as well. And then pass through that along the stream of the lipid phase, the turbulent form will flow following the packet number regarding the density and also the viscosity of the streams, and then that will determine how turbulent the flow is, and that will affect the production of the liposome itself. Now, because this flow, if you look at this one is in 2D. So basically, if it's in 3D, so there will be like a, depending on the shape of the entrance of that lipid phase, so that layer will form either in a smooth flat planar layer or will form in a basically in a circular. If you look at this in 2D, but through the front to the output of the device. So because of this kind of mixing is very irregular, the turbulence is something that we cannot control. So the yield itself will become variable depending on, as I said, the packlet number of whatever lipid phase and aqueous phase that you put into the device. Okay, so some very smart people, they decided, okay, this is something that we cannot abide with. This is something that is too much unpredictable. So they decided, instead of letting the stream mix by themselves, they decided, why not we control the mixing? So they come up with this thing called herringbone mixer. It looks like herringbone, even though I've never really, see the similarity, but it's called herringbone mixer. But it's fine. Anyway, the name is the name, it's up to the inventor of this product, calling it herringbone mixer. So this mixer, okay, it takes the job of actually mixing these two phases. So make sure that it is mixed within a very predictable method, very predictable procedure of how the stream will flow along the channel. So we can see that it's in action. Okay. For example, if you look at the diagram at the right, okay, at the first one, for example, at the entry of the channel B, the bottom one, the 0 cycles, and then you can see the 100 micrometers there. That is how the stream goes into the channel. So at first, you can see that the bright stream, two of them at either side of the dark stream, you can see that at the interface of it, there's a very light place where they start to interface with each other. So this is where the concentration gradient occurs, and this is where the liposome will form. Now, if you are considering something like a conventional microfluidic design, you follow through all this stream, the part where it interfaces will become considerably more turbulent, and then, you know, as I said, depends on the Becquerel number, that's how the turbulence will mix between these two phases. Now, the herringbone mixer takes care of this by actually churning the flow. You can see that at half cycle there, the first 1/3 of the channel is starting to be what's called like a warp, like they try to twist it, but only 1/3 of the channel, and then the rest 2/3 of the channel only starting to warp. But coming to one cycle, okay, it's immediately switches. The one initially the 1/3 of the side, 1/3 of the breadth of the channel suddenly changes to the 2/3 of the channel, and then becomes more properly mixed. So this mixing, this turbulent, this vortex can be controlled using this herringbone mixer. So this removes that uncertainty of the mixing. Therefore, you can see that in the diagram on the diagram on the left, one is no mixing, and then at 2 that is just laminar mixing only. But if you put in the herringbone mixer, now it starts to increase uniformly on how it mixes with each other. So every checkered board is actually doubles every time the cycle, they're moving down the channel, and then every time the cycle is increasing. So at 3 there, you can see that it's more than 2, and then progressively more and more mixed down the channel. But more importantly is that the vortex is intentional. It is intentionally mixed in a certain way so that we can be sure that at the end of this channel, initially we can see that at the end of this herringbone channel, they are properly mixed. So that's the important point. Right. Okay. All right. So now when we're talking about this evolution of microfluidics design, okay, over the years, even smarter people came out with even more ingenious design on how to do this microfluidics thing, right? So initially, a long time ago, people go for high energy techniques. We do for extrusion, for example, and then there were limitations in the application, difficult to reproduce, harsh process condition, because imagine you're trying to put through the liposomes through something like a sieve. Liposomes is very, very soft, and then it can easily break as well. And then it's not easy to scale up. What are you going to do? You're going to build a bigger sieve and then push through all the liquid that's going to require a lot of pressure as well. So it's going to be very difficult. Inline micromixing is basically the conventional design of the microfluidics, and then they become more and more refined. So microfluidic approaches, you go for the laminar flow and also the turbulent flow by the herringbone mixer. So in this case, there are some scaling challenges remain, because even if you look at the herringbone mixer, okay, the length of the channel allows complete mixing, but then again, that complete mixing, there are still some uncertainties for it, because yes, you can say that, oh yeah, the channels allow for the vortex mixture, but then again, this is something that also depends on the characteristic of the liquid that you put in as well. Higher and more viscous fluid become not easily mixed. So, people with ideas come out with, why not we mix this in glycerin, for example, way viscous. Can it be used for microfluidics? Maybe not. Maybe it can, but will it mix very well? Maybe not. So this becomes something that, the limitation of this herringbone mixer. And also that the mixers are very difficult to make. When I visited their laboratory in Vancouver, and that herringbone mixer, that striated line that you see in the channel itself, is something that is very, very small, and that it is also 3D printed. So that 3D printer itself has to have a very good resolution in order for it to not make any mistake in that making the mixer, making the ridges, the striations and things like that. So that's why I said that mixers are difficult to make. However, this shows that this thing can be scaled up, and it is transferable from bench scale all the way to GMP scale. So they come up with new things called this next gen microfluidics. So it's much easier to say, much easier to build apparently. So this one I actually haven't tried, but I have, back when I was in Vancouver, I actually have seen the test device, and I actually tried the device as well, even though during that time it was still a prototype, and he said that, yeah, you cannot tell this to the world. But I know I know that the code name for the development and everything, I stick to that. And then yeah, it is actually much faster to form, and also that it allows for much larger volume. And the advantage of this easy to make is not just saying that, oh, it's much easy to make through the 3D printer. It's not that, because when you are considering printing something on 3D printer or anything basically, the smaller the part, the striations, the ridges and everything, it is more susceptible to corrosion. Not in sense of that corrosion like a rust corrosion, but the basically the disruption of the flow and everything, because it chips out. The plastic itself has to be strong, and you have to remember this is in micron size. So basically it has to be very, very strong in order to not chip out due to the flow and everything in the channel. But the next gen microfluidics is just basically what it looks like. So it is much stronger, so that one chip can be used many, many times, and it allows for longer production time before it fails. Okay. Right. So I'll talk a bit about what I did in my project. So I used this thing. So basically, this device was produced by Precision Nano System, and then this one is the older one. The newer ones have seen that they have all the touch screens, all the very fancy things now. This one you have to connect with a laptop, and then you have to adjust the parameters of everything. Now this is just easy. It's just all touch screen, so much easier. But the basis of this device itself is just, if you look at it, it looks like it's just nothing. It's like a white box. Yeah, true. Because basically what it is two platform to push the syringe into the device, into the chip. So the more expensive device inside that device is actually the chip for the microfluidic chip. Now, what you do is that you put you have two syringe, one with the aqueous phase, the other one is with lipid phase. The lipid phase obviously containing all your lipids, and the aqueous phase containing drug, for example. Okay, in this case, you put inside this machine, and then you will start. And then the reason that it has this laptop connected to it is to control the flow rate and also the ratio. Now this is important because, as I said before, if you use something like thin film hydration, in order for you to control the size of the lipid, of the liposome, for example, is by post-processing. You extrude it, for example, the size of the extruder, okay, is what determines the outcome of your liposome. But in this case, what you have to control is during the process itself. You control the flow rate, so which means that how fast is the syringe going up, pushing the phases into the chip, and also how much one will move in relation to another. the syringes, because remember each syringes contain different phase. Yeah, right. So an example of the formulation that I did is containing the SPC, DPPC, and the SPG. Okay, all the lipid is like this. It has a very long name. Okay, you can Google for yourself, look what is. But yeah. Anyway, the good thing about using this liposome, lipid, and everything, you can always find a great combinations of lipids that you can use. There are lots of lipids. There are many things, lots of abbreviations, lots of acronyms and things like that. You can combine them in all manners in order to get one that you like for your formulation. And remember that the reason that I went into this research is actually to use old drugs with new carrier. So now this carrier is suitable for the drugs I want to use. If you go out and you want to have your own carrier, you can experiment it by yourself using your own combination of lipids and your own drugs. So that's the good thing about this. So there's someone that is actually a full-time formulation scientist, where his job is basically someone coming with a drug, a company, for example, I want this encapsulated. So his job is to find the best combination, the best ratio for each of the lipids, to get the release rate, to get the size, to get all the characteristics suitable for what the client wants. So that's his job basically, formulation scientist. Now, the aqueous phase in this case, I use the combination of irinotecan and cisplatin for the dual loaded nanoparticle. Okay, never mind that, we'll look at the result. But what I want to stress is the process itself, the process of making the carrier. The content, the payload is not as important. Actually. Well, important to a certain point, but not. Anyway, so we look at the characterization of this thing. So the reason that the formulation there is named 5, 6, and 7, every time I show this slide, people ask what happened to 1, 2, 3, and 4. Well, they didn't work, so that's why they are gone. There are also formulation 8, 9, 10, and the rest. They also didn't work. So I take the one that only works. So these are the ones. That's, as I said, you have to experiment with various types of lipids to find one that really suited to your needs. So this one suited to my needs. I mentioned the molar ratio, and this is the characteristic that come out from it. So that's good size, the size that I want, within 90 to 200 nanometers, good PDI as well. And then it's stable. So basically no changes, everything, yeah, stable. We did also some morphology study for the liposomes, but the thing about morphology study of liposome is this. If you look at the picture at the left, okay, that one, I think is something that people here know, is taken from SEM. Now, how do you visualize something that is very difficult to visualize? Okay, that's what I can say about it. Because these things are basically transparent. If you look at the liquid itself, it's very transparent. You can see the light will go through it. So basically what I'll do here, you have to suspend it, okay, and then you have to make it solid. Because SEM, you have to coat it, and then you have to do all the post-processing in order to see it under the SEM. Now, this was taken after I mixed the whole aqueous phase with more trehalose. Initially, the aqueous phase of the formulation also contained trehalose, but smaller amount in the inside, means that during the fabrication I use smaller amount of trehalose, and then after I purify it, I increase the amount of trehalose outside of the liposomes. There are some difficulties in this as well, because as you can imagine, the higher the amount of trehalose you put outside of the liposomes, okay, the more likely that water will come out from the liposome because of osmosis. Now, when this happens, the liposome will become smaller. So this has to be monitored using the zeta sizing. So I will have I did various concentration of trehalose, and then I see which one is affected by the amount of trehalose, and then which one is actually can be freeze dried. Because, for example, if let's say the concentration of 300 millimolar to 500 millimolar of trehalose is good for the size, means that the size doesn't change differently from, if you don't put any trehalose at all, means that the liposome can still maintain the water content and everything. But when you freeze dry it, because you want to put it under SAM and you freeze dry it, some of them might not be cryo protected enough. So some of them, when you rehydrate it back, you will find that the size change. So these are the things that you have to do in order to check whether your formulation will fit the need of what you want to do with that formulation itself. So for example, in this case, I found that between 400 to 450 is good enough to make sure that the trehalose protect the liposomes, so that during the imaging, we can see the supposed liposome in there. And also that when we rehydrated it back, we can get the same size of liposome from the solution. Okay. And the next one on the right is under the AFM, atomic force microscopy. So in this case, I think when I saw Dr. Awis's presentation just now, that reminded me a lot about the AFM, because that's how AFM works. It has a cantilever. So instead of hitting up the point, the cantilever is to a very, very fine point. I think like a 1 atom, something, 2 atom width at the top of the cantilever. And then it will detect a very subtle change in repulsion of the surface. For example, if you have a surface and then it moves along the surface, you can have a force mode, a tapping mode, or a contact mode. Contact mode means that the tip of the cantilever will touch the surface of what you want to analyze, and then it will check the tensile strength of it, this kind of characteristic of the surface. So at the back of the cantilever itself, there is a small mirror. There's a laser from the machine that will fire to the small mirror there, and then will reflect it back, and then the detector will detect the changes in the distance between the cantilever and also the detector. So that way they can detect bumps on the surface and this kind of thing. So this also proves a challenge, because for this to happen, you have to have your sample dry. So liposome really doesn't like dryness. If you put it on the flat surface and you dry it, it will become something like a jellyfish on the beach. Initially, it become like a spherical in the surface because of the surface tension, these kind of things. Okay, but when you dry it and put on the surface, it just lie flat. Now, when it lies flat, and then you measure the size of it, there is some contention on how do you want to basically say whether the size is correct or not. Because imagine jellyfish in the ocean, the diameter of it compared to jellyfish on the beach, they have different diameters because you lie it flat. So in this case, there are some problem in saying that, oh, this is the actual size. In this case, for example, you can see that there are some other bumps as well on the picture, but why didn't I select that? So the sampling becomes very selective, and it's just cherry picking between one another. So it's not really, I can say, as quantitative as the zeta sizer with the hydrodynamic sizing. Even though hydrodynamic has its own limitation in terms of it assumes the particle is spherical. It's not necessarily spherical, but it assumes so based on the Einstein-Stokes equation. But then again, we do what we have with what we can have. You cannot really count and size every single one of them, in situ. because you have like thousands and thousands of liposomes at once. But we do find some variations between these types of measurement in terms of sizing. For example, if you can look at the SEM there, actually some of the liposomes are covered by trehalose. The trehalose form like a continuous matrix connecting these like bridges between these liposomes. So if we say that, oh, that is the actual size of the liposomes, you might not be correct, because there are some liposomes. So there are some trehalose covering the liposome itself. So it'll be different. So what you can say for sure that it looks like that, and that's all there is to it. Okay. Right. So then we come to the purification. All right. So if we talk about purification of nanoparticles, the usual way that we do this is through usually centrifugation. Okay. You can go for like high-speed centrifugation, of which like you achieve like tens of thousands of Gs in order to have the pellets and the supernatant, and then you remove, you wash it again. But I found that usually these kind of methods, they are very stressful to the liposomes. Remember that the liposome is almost like a cell without the cytoskeleton. It's basically like that. So basically, if you put it under so much pressure, it will just flatten. It's not very strong structure. So the aim of this problem-solving exercise is to find a method of purification that is not stressful to the liposomes. You can scale it up, and also you can concentrate it. Okay, remember, because in centrifugation you also can concentrate the formulation. So this is what. One other options is to use filter centrifugation. Basically, it's also like centrifugation. Just like we put all the samples on top on the filter, and then we run in a centrifuge. So basically, all the non-liposome will fall down into the tube. Okay, it looks something like this, like a bottom there. You can see the centrifuge tubes. So there's a small filter in there, and then you put all the sample on top, you run on the centrifuge, and then all the excess liquid will fall down. Okay, but then again, this doesn't answer the first and foremost problem, which is how can we make this continuous? Means that how can we connect from the manufacturing side, the fabrication using the microfluidics, all the way to the purification process? Okay, so we come to the tangential flow filtration. So basically what you do is that instead of passing the fluid directly, following the flow of the solution, we also we make sure that the filtrate pass through tangential to the flow of the solution. Okay, this is using pressure as well, but the pressure is very low compared to what we experience with filter centrifugation. So it's not subject to limitations of dead end filtration. For example, the filter centrifugation. So it is more suited for the large-scale continuous manufacturing. Okay. So if you look at the diagram, okay, on the top right, okay, you can see that the diluent reservoir, the sample container, the peristaltic form, and then the tangential flow filtration device. What it does here is that the peristaltic pump will pump the fluid into the device, okay, and then in there, the force of the fluid pumped into the device will cause the pressure gauge there to increase up. So basically, we can measure the pressure inside the device itself. So this pressure will force the filtrate out from the device, but not the solution itself. So what happened here is that this solution will go back into the sample container. but some of the filtrate will have moved out from the device. And then this will continues on and on and on. If you can look at the past the pressure gauge there, there's like a small thing there, basically like a very narrow tip. So this one is the one that gives rise to the pressure inside the device itself. So the more you pump into the device, the lower the more you pump the device, but the outflow from the device is maintained constant. So you increase the pressure inside the device itself, and more filtrate will come out. Okay, so you will do this again and again, and then the diluent reservoir here is for it to go inside when the sample rise up a bit, so to replace the filtrate that comes out. So basically, this is kind of like rinsing the whole system. Okay, so usually the diluent reservoir here will be like 3 or 4 times the amount of the sample container, so that the sample itself will be rinsed and then go inside the device again, and continues on and on until all the diluent is finished, and then the sample itself can be concentrated. It means that the sample itself will run through the device until it reaches a point where there is a minimum volume that can be contained inside the device itself, of which that beyond below that volume there is no more filtration that will occur. So we did this during the comparison between the filter centrifugation, also the tangential flow filtration, using these parameters, and then we compare the results between these two. Okay, so look at the result here. Okay, for both of these method, okay, and this one is coming from the filter centrifugation, but for both of these results, actually for both of these methods, the complete removal of the unentrapped drugs is achieved at the 3 times of the sample volume from the diluent. So in case of the filter centrifugation, we actually manually put in the fresh diluent inside that, and in case of the tangential flow filtration, we do have the diluent reservoir there. But for tangential flow filtration, it takes 105 minutes to complete and can concentrate more, significantly more, compared to the filter centrifugation, which is much lower, and then you have to change it manually, and also at a much lower concentration factor. Okay, right. Sorry, I'll go back a bit about this. Now, the thing about the purification, this tangential flow filtration, is that it can be stacked up. For example, let's say that the diluent reservoir here takes up like 3 times of the sample container, and then it will have to go round and round and round for lots of time, takes like 105 minutes to complete, and so on and so forth. But you can connect these devices, like maybe 6, 8 of them, and then it will filtrate out much longer because of the surface itself. Surface for the filtration is increased, and then the microfluidic device can be connected directly to the sample container, and then the sample container can be directed out as well as the purified sample. So now we have 2 components within the continuous manufacturing process. Now we can connect the microfluidic device all the way to the purification device. Okay, right. Okay, so next one is the in vitro analysis. This is, as I said, the outcome of that payload that I've fit inside these liposomes. This is just to show that the formulation works. So basically that's it. Okay, so initially the idea is to combine irinotecan and cisplatin. So we do have different formulations from blank, which is anti-liposomes, to individually encapsulated irinotecan and cisplatin. So cisplatin in one liposome, irinotecan in another liposome, and one which contain both of them. The ratio of this drug is based on the synergisticity of irinotecan and cisplatin against A519 cells. So we compare that in vitro study. We compare these two between the free drugs and also the ones with the combinations. So now we use all three formulations for this dual loaded liposome with the irinotecan cisplatin. We can see that it is much more effective in killing the cells in A549. Okay, and also that because the initial ratio for the free drugs and the final drug final inside the formulation itself might be slightly different. So we look at each of the component of the drug separately. So that's why we have one bar for irinotecan and one bar for cisplatin. But then again, even if we take them individually for each drug, we can see that it's much more effective in killing the A519 cells. Okay, so that's a win there. But then again, as I said, the idea for the whole project is to get that continuous manufacturing side. Okay. Anyway, so for the conclusions of this is that we can see that microfluidics technique with the standard herringbone mixer provides a modular, rapid, and scalable irinotecan cisplatin dual loaded liposome fabrication method with the desired physical characteristics. So you can modify the characteristic based on what you put in and also the parameters for the microfluidic fabrication itself. And also that the tangential flow filtration method want to be more efficient in liposome purification, effective in the concentrating the liposome suspension, and also more suitable compared to filtration centrifugation for the large-scale operation. Now, the plan for the future for this is to actually get a complete product. Okay. This is actually a complete product. This is the combination of the cisplatin and irinotecan is such a product, and you can show that it's working in vitro and something like that. But then again, my interest in this is more towards creating the carrier itself. Okay, some carrier can be used universally. Some of them might be useful for some specific drug, and then the combination of the lipid and the combination of the drugs might give rise to something much better with much lower cost. So the idea here is low cost. Back in my university days, even some of my supervisors saying that, why not you find something like a new drug so you can encapsulate it and then you know, they become something new, everything. But why? It's not. For me, that will have lower reach to the people because some of them might not be able to afford the cost of that drug itself. But then again, the idea is to have the drug available to all people. That's why the topic for the talk today is nanoparticle for the masses. So everyone can have it. So we start with something small and then we scale up to something larger. Sure. Okay. I think that's all for me. Thank you.
Thank you, Dr. Taufik. Very nice and interesting finding. Yeah. Maybe any questions from floor? Any questions, especially from FarmTech?
Assalamualaikum, Dr. Taufik.
Waalaikumsalam.
Solah, yeah. So microfluidics is a technique to fabricate liposome from the beginning until purification, right?
Yes.
Is it correct? Okay. Other than liposomes, is it like solid lipid nanoparticles or nanostructured lipid carriers can be kita boleh buat menggunakan microfluidics?
Yeah, no problem. As long as you have two phases of anything, basically in this case aqueous and lipid phases, you put through that, you mix them together. So that's why I mentioned initially, microfluidics machine is actually a mixer. So anything that you need mix, in a very controlled, diffuse manner. So yes, you can use microfluidics.
Interesting. How much the bench top punya microfluidics tu, yang the most cheapest lah, the cheapest one lah.
Okay, for the as currently, I have contacted Precision Energy System previously. The one that I've used is called Benchtop. Now it's called Ignite. They have some rebranding. The smallest one is called Spark. That one, the yield is from 0.5 mil to 1 mil. So the Ignite one, the Benchtop that I've shown you, the yield is about 5 to 10 mil. Okay. There are larger ones all the way to the GMP parts, but if you're talking about the benchtop that I've used, the cost is 100,000 USD. The smaller one, the Spark one, is actually about 50,000 USD. The one that yields 0.5 mil to 1 mil. But actually, I have contacted Precision System, and they actually agreed to bring us the device here for demonstration, just for 2 weeks. Okay, but I haven't decided when yet, and I plan to extend that to 4 weeks so that we can have a small session, like a workshop or something, to show how it can be used, and we can just formulate whatever we want during those 4 weeks, and then we can have we just need to buy the microfluidic chips. So the chip itself is not that much expensive actually. So yeah, the device itself is expensive, and I don't think anyone in Malaysia has it. I have asked everyone. Actually, during the meeting itself with Precision Energy System, they do have an office in Singapore, and then the last time they have contacted someone from contacted them, but there's no news from whether they want to buy or not.
Any other questions? I have one question, Dr. Taufik.
Yeah.
How did you evaluate the purity of the purified nanoparticles?
How do you purify nanoparticles?
Yeah, because you are doing purification of nanoparticles, right?
Mm-hmm.
So how you evaluate the purity?
Okay. There is one batch that I do with a lot of liposomes at once. For example, I produce it using the same method again and again, so I yield something like 500 ml or something, a very large amount. Now, this large amount will have a specific amount of drug inside it. So I measure them and then look at the not negotiated, but the one that unpurified yet. So basically, I lyse the liposome, okay, and then look at the overall content. And then each of these large batch production, I will separate into the tangential flow filtration and also the filter centrifugation. Now for the filter centrifugation, what I did is, for one run, I will take the filtrate and then measure the how much drug will be left. Okay, inside that. Okay, and then give it a rinse, pour in empty diluent inside that, and run the centrifuge again and look. All right, and then at a certain point, the level of the drug will start to plateau, basically at 0, because you keep rinsing, right? So basically, at a certain point, it will just start to plateau, and that's when you already purified everything inside the filtration. The same thing with tangential flow filtration as well, except that I will vary the amount of diluent inside the that I use in the diluent reservoir. So 1 time volume, 2 time volume, 3 time volume. So basically, in the increasing amount of the volume, and then after each of the run until they are concentrated, I will take it and then measure the amount of the drug inside that. So at a certain point, it will plateau as well, because everything else outside of the liposome has been filtrated out of the solution. So the rest of that remain inside the solution that has been concentrated are the ones that I have lysed out from the liposomes.
Okay. Any questions? Others from others? If not, then thank you very much, Dr. Taufik. Thank you very much. Hope we can collaborate, especially for herbals, herbal extract.
Device tak ada lagi, Doktor. Device tak ada lagi.
Apa?
Device-nya tak ada lagi.
Bolehlah nanti minta duit.
Ada demo 4 minggu katanya.
I have one question, Dr. Taufik.
Yes.
Prof Tahir, Prof Tahir.
Ya, Prof.
Prof Tahir. Dia disconnect lagi.
Yes, actually, the most what called the effective way to produce liposome is using microfluidic technique, right? So, I mean, I want to know the how much this is a manual technique as well. Without this equipment, we can also make this what called liposome. So how much, I mean, the differences with regard to the quality, the topic between the manual preparation and if you compare with this equipment?
Manual, you mean like manual using microfluidics or manual using the thin film hydration?
Yes.
The thin film hydration.
Yeah.
Because when I did my project,
Hydration method, Dr. Taufik.
yeah, the film hydration method.
Yeah.
I went straight to the microfluidics because the aim of the project is to find something that can go continuously from production to the purification. So I didn't go through the thin film hydration because I don't know how to connect the thin film hydration to the purification method. The idea is about the processes, not the product. Sangkut lagi, Prof.
Mau disconnect lagi, Prof Tahir.
Boleh ke Starbucks lagi lah.
Okay lah. Okay lah, maybe can be continued ya, Prof Tahir.
Just little bit putus-putus.
Not little bit ya, long time putus. Boleh tak sama?
Maybe we have special discussion lah, face to face with Dr. Taufik.
Boleh.
Yes.
Okay lah.
Okay.
Okay lah, thank you very much, Dr. Taufik. A very nice presentation.
Level 5.
Okay, with that, then we will end our session today. Yeah, by Tasbih Kafarah and Surat Al-As. Okay, thank you very much, everyone.
Thank you very much. Assalamualaikum.
Thank you very much.
Assalamualaikum.
Salam.